atoms group here at Lawrence Berkeley Lab, but it's a collaboration between 4 different groups. Our group here at Lawrence Berkeley Lab, a group at Los Alamos Lab, a group at the University of Cambridge, and a group at Duke University. And so all of the work I will present today is truly a collaborative effort between all of the members of the Phoenix team. Everyone contributes to these together. It's a very, very collaborative project. So, presentation seems to have frozen, Peter.
It's frozen now? Yeah, it just moved right now. It's working right now.
Okay, thanks. Okay, so as Peter said, I'm going to speak about some new tools that we have in phenix.refine for dealing with refinement at low resolution. And why is refinement at low resolution a problem? Well, as you can see here, we have the phenix.refine target, which is a summation of the match to the X-ray data plus a weighted geometry term. And at high resolution, as you can see here on the left of my slide, the data looks great. We'd all like to see one angstrom data. You can see it's very easy to build a model into that either by hand or using automated tools. But as we move to lower resolution, here's an example at 3.8 angstrom, the density gets much less well resolved. It's very sausage-like, and often you can't tell exactly where the backbone trace is or where the side chains need to go. And so you need to include other information besides just the diffraction data to be able to build an accurate model. And so, of course, this isn't new. Knowledge from chemistry and biochemistry has been used for a long time in structural refinement, such as simple things like bond lengths and bond angles that you might get from Eggen-Huber or other parameterizations like that. Also knowledge such as non-crystallographic symmetry, which we can use to make chains that we know some relationship about, to make them look similar through refinement. We can use validation metrics, such as things that come from mole probity out of Dave and Jane's lab at Duke, such as acin, glyn, and histidine corrections, which we do automatically in Phoenix, and you can do in other programs as well as automatic rotamer correction. We know something about what side chains should generally look like, so you can use that information to inform your model building and refinement. We can use Ramachandran restraints, which have been used in COOT and also in Explore CNS. Secondary structure restraints, which have been used in CNS and other programs for doing Watson-Crick base pairing, for example, and also using information from related structures, such as Den refinement, which I'll speak a bit about later, that's been used in CNS from Axel Brunger's group, or methods in Buster and RefMac as well. And so today what I'm going to talk about are 5 new methods that we've introduced in Phoenix to do just this sort of thing, where you have low-resolution data and we want to see how we can use other outside information to improve the refinement of the working model. And so these are using reference model restraints in Phoenix, and then also torsion-based NCS restraints. Those two are related. And then also secondary structure restraints, Ramachandran restraints, and Den restraints. And the reference model, secondary structure, and Ramachandran restraints are all further detailed in a paper that will be coming out in Actor Chris D probably sometime next year. So first up, I'm going to speak about reference model restraints. And what do I mean by that? Well, here's a toy example where we have an aminotransferase structure, which was originally solved in the PDB at 3 angstrom, and then was later replaced by a higher resolution model solved to 2.3. And so you can see, if you look at the top, you have 2 chains that, if you just do a backbone trace, they align quite well. But if you then look, as I've split it out at the bottom, the low resolution model on the right has a lot more rotamer outliers. which you can see depicted in orange, compared to the higher resolution model, which just has a small handful. And so how can we use the information from the higher resolution model, which we are more confident in the model in where we place the atoms, to help us inform the refinement of the lower resolution model? And so what we do is combine two intuitions. The first being we know something about the rotameric state of the side chain. So if these models are very similar, whether it's the same protein or an abo form versus a ligandin form, or a similar protein from a different organism, we know generally what the rotameric state of the side chain should be if we solve this at high resolution. So what we can do is identify rotamer outliers in our low resolution working model, and if there's a good rotamer in the higher resolution reference model, we can just go ahead and fit what we think is a better rotamer in place of the outlier by just matching the chi angles. And as long as this still matches the density reasonably well and doesn't introduce significant steric clashes, this is likely going to be a better fit at that particular side chain position. So once we've done that, we've overcome some local minima, false minima restraints that would have been impeding the refinement. And then we're going to add reference torsion restraints to actually help steer the refinement and give it more information. And so what we do, we match all torsion angles between the working model and the corresponding angle in the reference model. And we do this through secondary structure matching alignment. So this allows us for the sequences to be somewhat different. We could still match the backbone angles and certain chi angles that match between non-matching side chain types. And then we're going to add all of these dihedral restraints to our energy target. And we put these restraints in what we call a top-out potential, which I've shown here depicted on the bottom right. And what this does is, instead of a normal harmonic, which is going to increase and restrain much more strongly as you move away from the target value, what the top-out does is, when you're very near to the target, it's going to pull on it like a harmonic normally would. But as you begin to move away, it will eventually top out or limit off. And what this allows is therefore to be automatic handling of differences between the reference model and the working model. And so if you really have a conformational change that's slightly different, or different side chains in an active site or something like that, that will be captured by this potential and will not actually pull those restraints to match if they're far away. And so in practice, what does this look like? The default that we cited is 15 degrees for a particular torsion. So here, the side chain on the left, all the angles are very close together and between the reference and the working model, and so those will all be restrained. Whereas on the right, you can see in this side chain, they're actually in two different rotamers, and so we don't want to just pull on that and twist it around really hard. It's going to not restrain that. And of course, if you want it to be restrained, you can set the default, set a higher default limit value, and then everything would be restrained. But in practice, this value seems to work pretty well. And so here's a couple examples of how this might work. You can see here on the left, this leucine residue. The starting position is in pink, which is a rotamer outlier. And so it's very hard for a refinement potential to fix on its own. So we correct it before refinement starts to match the good rotamer, which is depicted in green, and then following refinement with the reference torsion restraints, you end up with a better fit to the density and a better model, because now it's rotameric. And then similarly on the right, this glue residue, you end up getting it to match closer to the higher resolution model, which we have more information to support that as the actual position of that side chain. You end up with a slightly better fit to the density, and overall a much better model in the end, where we've decreased R free and the gap between R and R free, so we're overfitting less, and our mole probity statistics have all gone up significantly. So that's how it works in an example. How does it work in a real case? And this is a situation, a collaboration that we did here with a group in Texas, and there were three particular structures for this cyclic GMP-dependent protein kinase, where we had two different bound liganded forms and the apo form. And so the cyclic AMP bound form solved to 2.49 and was quite a nice model for that resolution range. And then the GMP bound version was much lower quality data set that only solved out to 3.2, and then an apo form to about 2.7. And so we use the cyclic AMP bound form as a reference to refine the other two structures. And what you can see here on the left is the mole probity validation for the highest resolution, the cyclic AMP bound form. And you can see it's a very good model. We see mostly green in the mole probity statistics. It's a low clash score, high Ramachandran favored, pretty low rotamer outliers, good bond geometry. So that's a good model. We can feel confident in that. And so using that as a reference, we go from the columns where you see a lot of red and yellow for both the GMP bound and apo form, where you have high clash scores, high rotamer outliers, not the greatest geometry. By using the reference model for refinement, we improve essentially all of the statistics, and we also lower R-free and decrease the gap between R and R-free. So we have a much better model, which we feel is, which statistically supports the X-ray data, and also geometrically makes a lot more sense. So these are the types of cases that we had in mind when we implemented these restraints to help improve your refinements when you can't get a higher resolution data set. Now, similarly, you can think of a reference model really in the same way as you think about NCS-related chains. It's another chain that we know should be similar, so it's sort of like extra crystal NCS. And so we thought, well, why can't we just apply the same torsion-based approach to restrain NCS-related copies? And so we did just that. We implemented the same limit-based pop-out function for NCS restraints. And this gives us flexibility where you can have real differences between NCS-related chains in your refinement, which is something you see if you do crystal packing or other considerations. There can be differences between those, and so you end up doing harm in a lot of cases by restraining them to be too exact. And we can also dampen the target, which means we're not going to pull on them to their average necessarily, but we're going to pull them towards each other. And if one of them is correct and one of them is wrong for that particular torsion, the data should hopefully guide, should hold more strongly onto the correct one. And eventually, after a number of rounds of refinement, they'll end up finding the correct area. And we can do this across multiple restraint groups, where you can, if you have 4 related copies and 2 of them are in one group and 2 should be in another group, that will all be determined automatically. And so you end up, it's real easy to parameterize this. And so automation was something we really wanted to keep in mind for this. And so users can specify NCS groups, but in practice it works really well to just let it automatically determine how to match the groups by sequence alignment, and then the flexibility of the restraint system will handle any differences automatically. And the restraints are constantly updated on the fly as refinement goes. So how does this work in practice? Here's an example of a 2.5 angstrom RNase S structure where there is areas that are significantly different, particularly this area around arginine 40 in the structure where you have 2 different rotamers for that particular arginine. And so if you just do strict Cartesian NCS, you can see here shown in pink on the bottom right where this arginine ends up from the A chain. It doesn't match its density at all. And the reason is because it gets pulled to match identically the B chain. And so you end up with a poor fit to the density and also other geometry distortion. And so using our torsion-based approach, you actually fit you're able to automatically detect that there's this difference, and so it fits that arginine correctly, leaves it where it should be, and then it also will then allow for improvement where there is a match. And so you get much better R values, as shown here in the table, and also a lower clash score and less rotamer outliers. So in practice, this works quite well and is available on the command line and in the GUI. So the next topic I wanted to discuss is secondary structure restraints. And so sometimes you don't have a reference model or you don't have NCS. You're working on something novel or you just can't find something that matches your model too well. And so how can we use other information from chemistry to help improve our refinement? And so we know what alpha helices should look like. We know what beta sheets should look like. We know what base pairing should look like. And so we can use that to generate hydrogen bond length restraints that are very similar to just the general bonds that we have already in the Phoenix geometry term. And we can do this for a full atom model when you have hydrogens, or a model where you don't, and then it will use the N to O distance in that particular case. And so we can automate the detection of these using a free version of DSSP, which is included in Phoenix. And this produces a very easy-to-read syntax, which the users could then edit if you either don't agree with how the annotation is done, or you would like to restrain something different based on what you know about your molecule. But you can see it's very easy to say what your selection is for a helix and what the sequence range, and then it will know which atoms need to be bound together to help pull that closer together. And similarly for beta sheets, you can select parallel or anti-parallel, and then what the sequence ranges are. And so it's very easy to do by hand if you want to do it that way, or often the automated annotation works quite well. Similarly for base pairing, we don't have DSSP, but we use Probe, which analyzes contact dots. It's a program from the Richardson lab at Duke. And we can identify all of the Sanger base sites in RNA and then the Watson-Crick pairing in DNA. And those are reduced to a similar syntax where we have the two bases that are to be restrained and what hydrogen bonding type those are to be used. And again, this is done all automatically just by analyzing your input structure, and in practice does a fairly good job, but you can edit it should you want different pairings. And this has been very successful in ribosome refinements and other large structures that are low resolution and a bit unwieldy to work with. And so often the differences are very subtle. You may not see huge differences in R, and that's to be expected because the data isn't very good. And so that's how you end up with a lot of these problems, is that the atoms can move a fair amount without any change in the residual. And so we need these other terms from what we know about chemistry to improve the refinement. And so you can see here is just an example. I've focused in on 3 base pairs in this particular structure, and you can see the density is just a bit of a mess, kind of a sea of density to fit that in. And so if you refine this without restraints, those bases will no longer be in the right plane relationship and may deviate a bit from each other. But by putting these bonds in, they'll move more in a concerted motion during refinement, so you end up with a better model at the end in terms of geometry validation.
Jeff, I have a quick question from somebody here.
Sure.
Are there any additional metrics in Phoenix right now that will allow you to kind of validate it as you including those restraints in addition to R3? Like mole probability has to be run externally right now, right, through the web server?
No, mole probability statistics actually are included. If you're using the GUI, at the end you get a validation window which gives you all of the mole probability validation. We've built it all directly in. And also, just doing Phoenix Refine in general, whether you're in the GUI or on the command line, at different steps of each macrocycle, a table will print which will give you a summary of Ramachandran, rotamer outliers, clash score, and the other geometry residuals. So that information is available to you through the course of the refinement as well.
Okay, thanks.
Okay, so the next topic is Ramachandran restraints. And this is somewhat of a controversial topic in that the Ramachandran restraint, the Ramachandran plot has traditionally been one of the key geometry metrics for evaluating your structure. So if your model matches this plot quite well, then you have a pretty good feeling that the overall topology of your fold is probably fairly accurate. But at extremely low resolution or very difficult cases where you have no other way to hold the model in place, these can be useful to go from very well hand-built structures to keep them in that position as you do refinement to try to help domains move or things of that nature. And so we have two potentials that we've included in Phoenix. This is work done in collaboration with Nat Eccles and Pavel Afanin here at LBL. The first is very similar to the potential that's used in COOT, which uses a modified 3D landscape where we've added a weak gradient for the flat regions of the Ramachandran plot. And then a more simple just distance-based metric, which comes from Thomas Oldfield's work, that just drives outliers to the nearest allowed point in the Ramachandran plot. And so here you can see what this landscape looks like similarly to what's used in COOT. And this is done in conjunction with Vincent Chen at Duke, where we've added these disallowed peaks, these high peaks in red that you see, and then we have beta sheet and alpha helix plots and other allowed regions within the space that we can then move along this potential in the energy term. And so this is for cases where things are you have them built really well, but there might be just some very slight problems. And here is an example where you can see in the original we have a Ramachandran outlier, and then after using Ramachandran restraints, there's a very slight turn. You can see you can see looking as you move back, each peptide just turns just slightly and still fits the density about the same, but you end up with better geometry at the end. This is the kind of case that we are able to capture with this type of restraint, but it can be extremely dangerous to use these, and it only should be used as a last resort. Because you're removing one of the key validation metrics when you do use this type of restraint, and often if your model is bad to begin with, you're going to just probably lock it into to another wrong position. It will just cram it into Ramachandran space that will look good on the plot, but doesn't make your model any better. It often can make your model worse. And so we don't recommend this other than as a last resort for a particularly difficult problem. And so just as the point to that, the first time we saw this used in publication was for this mediator head module in Nature this year, and it's a great paper. I'm not at all meaning to disparage the authors. It was a very difficult data set, and the model is really neat to look at. But you have to be careful if you do use a restraint like this. Saying in your paper what the final Ramachandran geometry is a fairly meaningless statement because, of course, your Ramachandran statistics are good. You've optimized to them. And so you've got to be careful in your methods saying that we did use these restraints, and so using that as a validation metric is no longer really a viable option. And so as you can see, here's an example of where problems can creep in. The helices and strands in this model look really good, but areas, loop regions, can be easily distorted using Ramachandran restraints. And here you can see each of these peptides I've shown in the Ramachandran plot all fit really, really well. If you just look at the plot, you would think that's probably a well-built model. But you can see for this loop there on the left, you have a lot of atomic clashes, and it doesn't really follow the trace of the density very well. So you have to be careful in using these and really look and see what it's doing to your model before you just go ahead and trust it. Okay, and so finally, I'm going to touch on den restraints, which is a new restraint that we're working on here in Phoenix in collaboration with Axel Prunger and Gunnar Schroeder. And so if if you've heard of this before, it's a method that Axel and Gunnar developed at Stanford, where you use, for very low resolution data, you can use information about the overall topology of the model to inform how it's refining in terms of domain shifts and large globular movements. So it's a bit more powerful than rigid body because it allows things to deform a bit, but it also keeps the model from going too far from the starting position locally while allowing global shifts to happen. And so this has been available in CNS, and we wanted to bring it to Phoenix to make it more easy to use and more widely available to users in general. And so briefly, what does DEN stand for? Well, it stands for Deformable Elastic Network. And what this does is you identify a subset of interatomic distances within your model, and usually this is between 3 and 15 angstrom, and within 10 residues in sequence space. And so you select some random number of these distances and restrain them during cycles of simulated annealing. And as you go, you can update the distance depending on a few parameters, which will allow those distances to change. So if the density is driving a distance to be different than what the initial restraint is, it can, depending on how you've parameterized it, allow for that to deform and adapt to what the data is saying that distance should be. And then over a number of cycles, this allows for large motions of domains relative to each other and things like that. And so we run this repeatedly over a number of cycles, and you can end up with a better model at the end. And these reference distances can either come from a higher resolution model, or if that's available, or a homology model, or just even the starting model if that's all that's available. And so this is very new. It's not yet available in the Phoenix GUI, but it is available on the command line if you are a command line warrior and are willing to try that out. And so just as an example, comparing this between CNS and Phoenix, for one of the cases that was studied in Axel and Gunnar's original paper, you can see that using the Cartesian-based simulated annealing method in Phoenix, we're able to essentially replicate the results that DEN in CNS was able to do, and then using our torsion-based approach, we're able to do a little bit better. And this has been something that we're still actively working on. There are cases where, in most cases, they perform about the same if you're able to optimize them. And that's the real trick to doing DEN, if you've tried this before in CNS, is that you really need to optimize the weight and gamma parameter, which controls how easily the network is allowed to deform. And so you need to try a bunch of different combinations. And so this is the one limitation of the method, that's very computationally intensive. But we're trying to come up with ways to improve that, and if you have access to a lot of cores, for example, then this is easy to parallelize in Phoenix. And so the more cores you have, the more combinations you can try in the same amount of time. And so it's definitely a good model for SP Grid with the available computational resources to be able to do that. And so we hope to come up with ways to improve that, but that's where things stand at the moment. And finally, I'll just add that here's a visual depiction of what this network looks like for this particular example, and you can see that there are areas that don't have restraint, and that's okay. The reference model does not need to cover the entire working model. You can match just one domain or part of a domain, or truncate things if you're only confident about the core or something. These can definitely still help the refinement no matter how much information you give it. And with that, I would just like to acknowledge all the members of the Phoenix team and all of our various collaborators. Of course, our funding, which comes from NIH and the Phoenix Industrial Consortium. And at this point, I'm happy to answer any of your questions.
Thank you very much, Jeff. And I have a couple questions here for you from Tirumala. he's asking at what stage of the refinement one should apply those knowledge-based restraints. And in their lab, they typically tend to do it towards the end when the refinement is kind of converged and they're fixing rotamers. Would you worry about it throughout the process, or what is your recommendation here?
I think it very much depends on the situation. If the refinement is going well without any outside information, then at the point that it kind of stalls out, like you said, that could be a way to then get you to the final answer. But the danger is, on the flip side of that, is that you may be refining your model into a difficult false minima for it to get out of. So it's also, I think, would cut down on the overall amount of refinement time that you need to finish your model if you start with knowledge-based restraints from the beginning. If you've done a molecular replacement, or if you have a pretty good starting model, it may help you get to the final answer more quickly if you start with some of these restraints, particularly with the reference model or torsion NCS, because they're so flexible. There's no harm in using them from the outset to help you.
Yeah, and from our experience, I think with the same comment, we try, especially with NCS restraints, if they can be kind of applied automatically with your new approach, that could be much more powerful and saves you quite a bit of modeling.
Yeah, I think in general that does work best.
Yeah. I don't know, because you said you could potentially fire up Phoenix on your computer and show it up. I'm not sure if people are familiar with those tools, or if any of, would you like to see any of the interfaces, how those things are triggered? Just send me a text message if you would like to do it, and I'll ask Jeff to maybe fire up, unless you guys already are familiar with the interface, then there wouldn't be any need to do it now. So, please send me. Yeah. I don't see any text messages popping up. So, okay, yes, one person is asking at least for a demo. So if you can.
Okay.
Maybe. So we're done with our 30-minute quick coffee break. So if you guys get enough, you're welcome to just disconnect, and we'll just go for a couple-minute demo right now about Phoenix. You're obviously welcome to stay. And again, if you have any other questions, please send me in the chat window, and I'll relay them to Jeff. Yeah, Jeff, so you can.
Yeah, well, I'm just pulling up the app. Give me 1 second.
Okay. And again, our next presentation will be on January 12th with Vladek Minor on HKL 2000 package. And if you have any general comments or even questions for Vladek ahead of his presentations, you're welcome to send me an email or send an email to Michelle, and we can ask Vladek to answer those specific topics ahead of his presentation.
Okay. Are you able to see the Phoenix refinement?
Yeah, I can see your.
So once you're in the GUI and you want to run any of these methods, for example, we might have this RNase-S model, and so just as an example, let's say we have that. We've read in our model, and we've read in our X-ray data, and then this model has NCS, so I'll show you how to turn that on. But let's say, just as an example, if we had a reference model, you would add it right here, and so we would add another file, and let's say perhaps it's from a different round of refinement or from a different model. And here, so now we've read in 2 PDB files, and you can see they both say input model. You want to highlight the one that's your reference model that you want to use for reference restraints, and then right-click over here on data type, and you can see you can change it, what its input type is. And so in this case, we could say reference model. And then if we go to refinement settings, we would need to then turn the reference model restraints on here under restraints and weighting. And so if we select that, we'll get a pop-up window with a number of options we can set. And so the defaults usually are pretty good for the sigma and limit, but if you have a particular reason that you want a tighter limit or a bigger radius convergence, you can change that there. And then you can also do some play around with the alignment and things like that, and we can also shut these off automatically for NCS. And so we have a very complex model where some of the chains are unique to that particular model, and you have a reference for that. We may want to restrain that, but not other chains that have NCS-related copies. We may want to let the NCS take priority for those particular torsion values, and so we can shut that off here if we so chose. But in most cases, the general refinements, the general settings here for reference model are quite good, and so we can say okay for that. And then if we want to do NCS, we can turn on the NCS restraints, and then here by clicking NCS restraints, and then here we can define the type. And so currently the default is global, which is a Cartesian-based method, but if we want to use the torsion-based approach, we'll change this here to torsion angle. And then if you go to the settings menu and look in all the different settings, we have more control over the reference model, and we can also have more control over the NCS. And so I probably should have said this before, but we can specify if you don't trust the automatic detection of reference groups for reference model, or the detection isn't working correctly, you can specify reference groups. And so perhaps in the reference model, the chain that you want to use as a reference is chain B, and so we would say chain B. And then maybe we want to apply the restraints in chain B to chain A, or maybe it's also chain B, or it could be both of them. We can apply it like that, and so that gives you a lot of flexibility with how you're inputting your reference model. If you have your reference as a single chain and you want it to apply to 4 copies in your model, you can do that with these reference group selections. And the hope is to have that all work automatically eventually, but there's still some cases that it doesn't detect automatically, and so you can easily do that with these sorts of reference group selections that you might be familiar with from doing selections in other parts of Phoenix. And so that would get that going, and then in terms of NCS, you can do the same sort of thing where we define NCS groups. You can add a selection for what we want to match with NCS, and it would be the same thing.
And is this working equally well for protein, DNA, RNA, sugars?
Definitely for protein and RNA. Sugars are a tough beast. So those I don't think will necessarily work correctly right now, or ligands for that example. It does focus just on protein and RNA. But sugars is an active area of development here in our group at LBL. Just in the past week, we've been thinking a lot about a few different things related to sugars, and so Nigel is working on a program called CarboLoad, which will help to build restraints more easily for glycosylated amino acids and other bound sugar situations to help improve that. But this does work quite well for proteins and RNA, DNA in conjunction. And you can also do, you can change the NCS options, same sort of thing as the reference model, where you can have a different limit parameter. And so this lets you customize your refinement if you want the NCS to be stronger than the reference model, but still want to use both. You could do that. You can also change how the B factors are being affected by the NCS relationships and things like that. So that's all controllable right here. Of the other methods that I talked about for doing secondary structure restraints, you can turn these on here by selecting the secondary structure restraints, and then by default it's going to do everything automatically, but you can set it to no, and then either you can supply a parameter file with all of your generated restraints, which you may have come up with from by running the tool on the command line, and then or you can define them here in the GUI. But maybe you only want to do helices, or you just want to do sheets, so you can turn things on and off and play around with the distances. The defaults really do work pretty well. If you're new to the method, it's a good approach just to try that and see what it does. And if you're not happy with it, then tweak some of these parameters, or we're happy to work with you to find out what the problem is. And so, as an example of what that looks like, bigger. So if you're on the command line, you can run phenix.secondary structure restraints and then pass it a PDB file. Run that, it's going to give you a whole bunch of selections. And so here you can, I hope you can see that, but you can see this defined a number of different sheets based on the chain and res-seq selections, and also helices. And so you can save this output from running the command on the command line. You can save this in a parameter file, and if you pass that to read that in as another option during your refinement, you can use that to define them ahead of time. This is what Phenix would do automatically. But more importantly, if you look at this and then analyze it in what you're looking at your model, you may disagree with some of the selections, or want to augment it with some other strands that maybe didn't get identified by DSSP, but you are pretty confident that that's part of the same sheet. You can then edit these by hand in a text file. And we're working on ways to make this more easy to do in a GUI-based setting. But that is not entirely complete yet, but hopefully we'll make that as intuitive as possible. And then lastly, if you do want to turn on Ramachandran restraints, you would go to this PDB interpretation button. We've inherently hidden this to make it a little bit more difficult to turn on. But you would come in here, and then under peptide link settings, there's a Ramachandran restraint button. And so you'll click on that. And now, if I were to start this refinement with the Ramachandran restraints active, because this is a 2.5 angstrom data set, it's going to throw a warning message and tell me that we don't recommend doing this below 3 angstrom. Are you sure you want to continue? And so you can override that and then continue with the job. But we tried to make it a bit more careful. One thing that I would recommend is we find a lot of users will, if they're dealing with a difficult structure, will just come in here and turn everything on at once. It may work, but if it doesn't work, it's hard to know what the problem was, because you've tried so many different things simultaneously that may be fighting against each other, particularly if you're starting with bad geometry. And so what I would recommend is try these tools initially, just one at a time, or in small subsets, and run a refinement and see how it goes. And if it seems to be helping your model, your validation getting better, the R values are moving in the right direction, then go along with that, and then you can add more restraints as you go. But it isn't necessarily going to give you a better model by just turning on all of the options that we have available. You should try to use them in situations where you think it might help. You know you have NCS. Start with the NCS restraints and see how that helps your refinement. And then you can add secondary structure restraints later if it's still not working very well, or things like that. So I would say use these as you go, but don't necessarily just try everything at once. And then I guess finally, the den restraints are not yet available here in the GUI, and they're a bit difficult to get set up entirely right now, and it's very fluid from version to version as I've been developing it on how to get those to go. So if you're in a real hurry to want to use them in Phoenix right away, you're welcome to email me, and I will help you show you which version to use and show you how to set up that refinement. But hopefully within, certainly by the next release, these will be available in the GUI and much easier to turn on.
Okay. And you guys have the software support to SB Grid. We typically update Phoenix on a regular basis, so you always have access to the newest releases. And
Okay.
also for Dan, we remember we had the webinar, I think, a couple months ago, Daniel Donovan. We also, if you wanted to check more weights and run it on the grid, we have facilities for this, so you can just email us to ask for access.
Great. That's good to know.
Option. So Jeff, any places where you guys will be, Phoenix will be presenting in the near future? If people go to conferences, workshops, any venues that you would advise people if they wanted to learn more about it then, or contact you guys? I know there is developer workshop coming up, but that's restricted to the Phoenix developers, right? Or
yeah, and we won't be doing a user workshop at that particular.
Okay.
But we will certainly be doing. A lot of us will be at the Methods Gordon conference this summer.
Okay.
Bates College in Maine, the refraction methods. So that'll be a place to interact with the group, and we should be doing some workshops at the ACA meeting, which follows that in Boston, July. So that's in terms of big meetings. That's what I know of so far. We're doing a 3-day workshop in Mexico, so if there are any SP Grid users down that way, that's coming up in April.
Okay.
I don't know of anything else offhand yet, but
that's plenty.
but there's
right.
likely to be more through the course of the year.
Okay, and you guys can always contact them directly or through the mailing list or email addresses.
Yes, please feel free to contact us through the message list, or you're welcome to email me directly if you have specific questions. But we try to get reply to questions within hours, if not minutes. And often if there's a bug or a feature that's highly requested, we can make changes within a day. So it's a very fluid process, but we try our best to be in touch with the users and actively work with them to make the software work better for the problems that they're dealing with.
Thank you very much, Jeff. Thank you, everybody, for participation, and we'll see you guys after holidays on January 12th.
Great. Thanks very much.
Thank you. Bye.
Bye.